I want to thank the organizers. Thank you, David, for this kind introduction. And I want to thank the organizers for this meeting. It's wonderful to see that you have this program on IVRI. I think it's timely, and I can't think of too many places better than this to have a program on immunology, infectious diseases, and cancer. So what I'm going to do today is discuss two things. One, I'll discuss some data that we've already published, but I'll show you a little bit of new data on that, which is on combination therapy using PD-1 blockade and IL-2. This will be the first third of my talk. And then then most of my talk really will be on understanding the differentiation of human memory CD8 T cells. And pretty much all of this is unpublished data. So let me start with a slide that many of you may have seen before, but it kind of summarizes work that we and many other people working with chronic viral infections have shown. So basically, after an acute viral infection, one generates a very large pool of effector CD8 T cells, which contribute greatly to the control of the viral infection. After the virus is cleared, about 95% of these cells will undergo apoptosis. And this is a good thing because during this phase, you can get up to 100,000-fold expansion of antigen-specific cells. You need to cull the numbers down. So only about 1 to 5% of these cells survive and then give rise to the pool of highly functional memory cells. This slide is an oversimplification of functional memory. Of course, there are many different subsets of memory cells, but the message I want to give from this slide is that after an acute viral infection is controlled, you have highly functional memory cells both in the lymphoid and in the non-lymphoid tissues that can respond very rapidly upon reinfection. But what we discovered using the mouse model of chronic LCMV infection is that in contrast to this good scenario that happens following an acute infection, if the mice are chronically infected, then you get actually functional exhaustion of the virus-specific cells. The cells are physically present, but they're no longer functional. And this observation was made by Alan Zayak in a paper that we published in 1998. Others working with this model also came up with the same observation. So the discovery that the T cells are physically present, but they're not functional, of course, this raises interesting possibilities of rescuing function back into these T cells, as opposed to the T cells were deleted. Work that was being done by many other groups showed that also in cancer mouse models, you have TILs. They are there, tumor-specific are present, but not very functional. And these findings, which were made by us and the people studying cancer models, were very quickly extended in many human viral infections. People infected with HIV, hepatitis B, hepatitis C viruses. Again, these are systemic, ongoing chronic infections with continuous stimulation of the T cell receptor, again, results in various stages of T cell dysfunction. And of course, the cancer human studies, and you heard this wonderful talk by Jim describing that in human cancer. When John Very came to the lab, we did gene expression profiling of these exhausted cells. And we found that among the many changes in the gene expression, the expression of the gene expression as well as the phenotypic markers, a characteristic feature of these exhausted cells was high expression of many inhibitory receptors. And among these were CTLA4, TIM3, PD1, LAG3, 2B4. Many of these receptors that regulate T cell function were very highly expressed in the exhausted cells. And I'll tell a little story about my collaboration with Jim many years ago. So when we got the gene expression data and CTLA4 was high, PD1 was high. I have to admit that at that time I did not know what PD1 was. Okay? But of course I knew what CTLA4 was. And so I called up Jim and I said, Jim, can we get some of your CTLA4 antibody and see if we can get some rescue of the exhausted cells? And interestingly, CTLA4, at least in this model, had no effect, had zero effect. It was like injecting PBS into these mice. Okay? So I called up Jim and I said, Jim, this thing doesn't work in our chronic LCMV model. So Jim said, well, he went over how he did it. And I said, he had sent us the antibody, we put it in the freezer or somewhere. He said, well, you probably got messed up. What you do is you didn't really store it properly. So I'm going to send you another batch, and you plan it so the day you get the antibody, you put it into the mice. Okay? So we planted the mice were ready, the antibody arrived, we put it in, again, no effect in this model. And then we said, well, what is this thing called PD-1? Okay, and so, PD-1 I read up. Tasuku Honjo had cloned PD-1 many years back. I was totally unaware of what PD-1 was. This is going back way in the early 2000 when we were doing these studies. And then I found out that my very good friends, Gordon Freeman, who actually I was a graduate student with, had done a lot of wonderful work on PD-1. He had cloned PD-L1, had antibodies that block PD-1, PD-L1 interaction. So we got antibody from Gordon Freeman and did the experiment with anti-PD-1 blockade. And we found that actually this was quite striking. There was an immediate expansion of these cells, in some cases up to 10 to 50-fold, but the cells expanded. But more importantly, they became more functional. So there was expansion followed by increased function, and that resulted in reduction of the viral load. Of course, these cells did not become as functional as these cells, but they moved in the right direction. And in that and this was work done by Dan Barber, who was a graduate student in the lab at that time. And we also did the combination. It's kind of interesting. So we tried, of course, the combination with the CTLA-4 and PD-1. And again, that had no synergy. And that data actually was published in this paper, but is in a supplementary figure. So I think nobody knows that data. So in the viral, it's very interesting. I point this out because of the potential similarities between cancer models and chronic infection models, which is PD-1 working in both. But there are also some interesting differences. And I think we haven't dissected why CTLA-4 doesn't work that well in the system. But perhaps there's something to learn there in terms of looking into that more deeply, in terms of why in this chronic infection model we didn't see any effect by CTLA-4 monotherapy or in combination. And then there was work done in subsequent years by Rama Amara at the Emory Vaccine Center showing that PD-1 works in SIV-infected monkeys. Not a dramatic effect, but certainly a movement in the right direction. And then a group at the NIH did anti-CTLA-4 in the SIV infection model, and again found no effect. So it looks like at least in a few viral models where CTLA-4 has been used, the effects have not been that striking, either by singly or in combination. And to me, this is telling us perhaps something very fundamental about how CTLA-4 works. And I go back to the model actually that Jim has about maybe it's priming and it's kind of broadening the response. And that perhaps is likely to have more of an impact on tumor-specific cells because you're seeing whereas in a viral model, where really you know the genes, you define it's a small genome, you know what the epitopes are, and perhaps that little bit of an epitope spreading that might be happening might be very minimal. It's speculation at this stage, but I just want to leave you with this message that there's probably something to learn also when you find a difference between a chronic infection model and a viral model, even though the characteristics of the T cell actually are very similar, but other mechanisms might be quite distinct. So what I'll transition here to is combination. And Jim actually gave a wonderful overview of the many, many combinations which are happening in cancer, not only cancer models, but also in the clinic. In fact, already approved treatment with the IP and the anti-PD-1 blockers, which is showing actually wonderful results. Okay. So in our lab with the LCMV model, over the years, we've been playing with many different combinations. We've tried it in combination with other inhibitor receptors and seen some effects. We've also tried with therapeutic vaccination, showing actually there's wonderful synergy between therapeutic vaccination and anti-PD-1 blockade. But of the 10 plus things that we have tried, the one that's given us the most striking results or the most striking synergy is IL-2, low-dose IL-2 plus anti-PD-1. And I'll share with you a few of the slides from published paper on this, showing you this remarkable synergy in this combination, and then show you a couple of sets of new data which might provide some insight into why we are seeing this really remarkable synergy between IL-2 and PD-1 blockade. So here is the protocol. So we have a mouse that's chronically infected with LCMV. And then it's a 2-week regimen where we give either anti-PD-L1 or anti-PD-1. We see the same result, at least in this model, if we use anti-PD, whether we block the ligand or we block the receptor, very similar results are seen. And then at the same time, a low-dose IL-2 was given. This is the collaboration with Kendall Smith. And then 2 weeks later, we analyzed the virus-specific T cell response and the viral load. Okay? It's a very simple experiment. Daily IL-2 and then blockade in this window by 3 or 4 injections of the anti-PD-1. Okay? And the results were very striking. And this was work done by Erin West, who was a graduate student in the lab and was published in 2013. Okay? So this example shows you looking in the PBMC of the same mouse, pre-treatment, day 8 post-treatment, day 14 post-treatment. So this is the control mouse. These are your tetramer-positive cells. And if you look at function of these will be highly dysfunctional. IL-2 by itself does expand antigen-specific cells. Here is anti-PD-L1. Again, you get this expansion that we and others have shown before. But look at this synergy. This is a ridiculous number of antigen-specific cells. Okay? And that's a single epitope. If there are other epitopes that also expand. In fact, huge, almost 50-60% of the CD8 T cells now are specific for this, within this 2-week treatment of IL-2 plus anti-PD-1. Yeah. And this is happening not only in the blood, but if you look in the tissues. Here are the tissues. So this is killing the mice after that 2-week treatment and looking either in the spleen, lung, liver, or bone marrow. Let's go across this way. So these are the 4 groups: the untreated group, IL-2 only, anti-PD-L1 blockade only, and the combination. So this is about 1% of the CD8 T cells are specific for this epitope. Here. And again, most of these will be dysfunctional. IL-2 increases it somewhat. PD-L1 increases it, but again, this huge expansion. Yeah. Same thing is seen in the lung, very large expansion. In this case, PD-1 also is very good by itself. Yeah. Liver, again, this huge expansion. Bone marrow, huge expansion. So there's a systemic expansion of a very large number of antigen-specific CD8 T cells in this co-treatment. And not all of these cells are functional, but now almost 30, 40, as opposed to only less than 1% or so making interferon gamma and TNF-alpha. Now 25 to 30% of these cells will score as being polyfunctional in vitro, in your in vitro assays. And this is accompanied by, this is just one of the many experiments we have done, looking at viral titer. This is in the serum. The tissues show even more dramatic effect in many cases. Here's PBS group, IL-2, some effect, PD-L1 much better, but here almost complete clearance from the serum in the majority of the mice. This is one of many experiments done. So the question is that why? Why are we getting this striking synergy? Okay? And we've gotten a clue from something that was recently done by a postdoc in the lab, Masao, where we did, although this project had been there, it was a sitting. Masao came to the lab a couple of years ago, and we picked up this project again. So we did something very simple. We did RNA-seq and looked at the gene expression profiling of antigen-specific cells following these 4 treatments. And the next slide is a summary of that. Don't look at the slide yet. I'll give you I hate showing slides like that. Okay? So ignore the slide. Listen to me, and then I'll point out a few things. Okay? I hate these gene expression slides. Okay? So what that slide shows is that if you look at the gene expression profiling of an exhausted cell, it has a certain signature. If you do anti-PD-1 blockade and then look 2 weeks later, it's actually not that different. Okay? Of course, since the cells are proliferating more, some of the proliferative genes are proliferating more. But in general, it's not a very different cell, pre and post blockade. Okay? And interestingly, a characteristic, if you do the genomic analysis and look at what signatures are, an interesting characteristic of this is that the PD-1 blockade, so this is here, this is pre and post, okay, pre and post, some differences. This is the untreated group, and this is after the PD-1 blockade. Now remember, you have many more cells. They've undergone proliferation. They're also slightly more functional. But you look at this is, of course, cherry-picking some genes over here, but they're very similar. And what these genes represent over here that we're looking at are genes involved in cell death. Okay? So many of the genes involved in cell death were there before in that population. And after the blockade, even though you've gotten a very beneficial effect, again, it still has a characteristic cell death signature. Okay? But look at the striking change if you do IL-2 alone Now, very different gene expression profiling here of the IL-2 alone treated, and interestingly, also of the combo. So this signature is the dominant signature also in the combo. And what this represents is that all of this, basically all of these genes involved in cell death are very high pre-treatment, post-treatment PD-L1, but when you do IL-2, these are repressed. So it's repressing many of the cell death signature genes, okay? And the combinations give the same thing. And then the genes which are upregulated selectively by IL-2 alone and combo alone are involved in this thing that's called proliferation, but basically it means cell survival. Okay? So it's a very interesting finding that the IL-2 plus PD-1 combination is now giving you cells which have much more durability. Okay? And this gets back to an interesting point that Jim made in his talk, is that the durability of some of these treatments is perhaps not that long-term. Okay? And so this might be one way, this combination might give us cells which are much more durable. Okay? And I'll just show you one phenotypic analysis slide which will tell you that really some interesting thing is happening here. So one of the many genes which are selectively upregulated by either the combination treatment or IL-2 alone is CD127. Okay? So CD127 is the receptor for IL-7, and IL-7 signals are essential for cell survival. In fact, work that Sue Keck did in our lab many years ago showed that this is a canonical marker for the identifying long-lived memory cells. Okay? So because they need this IL-7 signaling for long-term maintenance. Okay? So if you look at IL-7 signature expression, here are the exhausted cells, and they express very low levels of CD127. In fact, all of them are sitting over here. After IL-2 treatment alone, now you have a subset that expresses IL-2. Anti-PD-L1 alone, even though these cells are more functional by the criteria, no CD127. That is, this looks exactly like this, okay? And this is the combination. Okay? So I think that what we are finding, and of course we'll do more experiments, do a little bit more in-depth analysis of potential mechanisms in terms of signaling. We are collaborating with Warren Leonard's group in this project to understand really how IL-2 signaling is changing the gene expression program of the exhausted cells. Okay? What's also interesting is that and there's a little bit of a counterintuitive observation here with IL-7, because if you take an effector cell at the early stage and add IL-2, in fact, CD127 goes down even more. Okay? But what we are finding with the program of the exhausted cell is a little bit different than what one sees with an early activated cell. Okay? So many of these are very cell context dependent in terms of how the cytokines act. And so this is an area that we will explore in collaboration with Warren's lab more mechanistically, but I leave you with this message over here, is that this combination, the reason for the large number of cells is because there's more survival. When you rescue cells by PD-1 blockade, many of the expanded cells are still dying. And here what we are giving them is a survival signal that's coming from this combination treatment. Okay? And so basically increased cell survival and durability of the T-cell response really addresses the question that Jim brought up in his talk about the durability of responses after PD-1 treatment. Okay? And so this might be a combination that would be worth testing in the clinic. Okay? All right, so now I'm going to switch, and the durability is a good transition to my next talk, which is human CD8 T-cell memory. About 10 or 15 years back, we switched from working with mice into humans, CD8 T-cell responses, and now about two-thirds of our lab about half our lab actually works on human T and B cell responses. Okay? So to look at CD8 T-cell memory differentiation in humans, our interest really was when we got into this human T-cell area, CD8 T-cell area, wonderful work had been done by many, many people working on chronic viral infections with HIV, CMV, EBV, so on. But there was very little information about CD8 T-cell responses after an acute infection. And in particular, we were interested in looking at a primary CD8 T-cell response, not a recall response. We wanted to study the response from the initial generation to the long-term effector generation memories. We wanted to look in a population where we could look at a primary CD8 response after an acute viral infection. So how do you do this? It turns out actually there's a wonderful system for studying this, which is the yellow fever virus vaccine. So the yellow fever virus vaccine was developed by Max Titer in 1937 What he did was he took a virulent strain called the ACB strain. ACB actually is the name of the patient who we got the virus from. And then he passaged the ACB strain in tissue culture on many, many times in an empirical way. While he was doing the passages at different time points, he was taking the isolate and testing its virulence in monkeys. The CB strain is highly virulent in monkeys, and then he ended up finding a strain which he named the YFP17D, which actually had the right balance. It replicated enough to induce a good response in the monkeys, but did not make the monkeys sick, and all of them are protected. This then very quickly became a vaccine that was licensed and gives one of the best vaccines ever developed, and Max Titer was given the Nobel Prize for this in 1951. The first Nobel Prize given for any vaccine development was given to Max Titer for the YFP17D vaccine. So this is an acute infection, live attenuated virus acute infection. It elicits both long-term T and B cell responses. Protective immunity in this system is predominantly due to having very high levels of effective neutralizing antibody. But this vaccine also generates very good long-lived CD8 T-cell memory responses. So this is the system that we have used to study human CD8 T-cell responses after an acute viral infection. And a couple of papers were published by us. Actually, first paper was in 2007, then a paper by Rama Kondi in 2009, where Rama identified a CD8 T-cell epitope from one of the non-structural proteins of yellow fever virus. This is presented by HLA-A2, and is a wonderful epitope. It's generated, it's a dominant epitope, generated high frequencies by anyone who's HLA-A2 positive. So almost every one of those, about 30% of our population is HLA-A2 positive. So very easy to do clinical trials in these individuals. All of the individuals that we enroll in the study are naive to yellow fever. We do a screen on them before. And since yellow fever is not endemic in North America, basically there's no issue of exposure here. So we can look at a primary infection, and by using this tetramer, you can track antigen-specific responses for a long time. And here's an example of this thing. So at day 14, about 1% of CD8 T cells are specific for this NS4B epitope. And this is all in the blood. And it peaks around day 21, so you have up to 2%. And then you can follow this. This is followed out to day 154. But actually, you can follow it out to 2 years, 4 years. In fact, I'll show you some data even 10 years later. You can still find this at low frequency, but you can still find it. Okay? And this is just showing you the kinetics of this response. Okay? This is the viral titer during the acute infection. And actually, in a study that was done several years back that we did, we actually sampled the virus every other day in the blood and did a quantitative PCR. So the virus peaks around day 7, day 8 in most individuals, okay? And then is gone from the blood by about day 15 or so. So this is the peak viremia, and then the cells are maintained for a long time. Okay? So we now had a system where we could study antigen-specific cells for extended periods. So we could do many things. We could look at gene expression profiling of these cells from the blood. We could look at phenotype and so on. Okay? And so I'll share with you a few of the things that we've done. Okay? So we've asked 3 questions, and I'll show you data on 2 of them. The first question we asked was, or one of the questions we asked, what is the rate of homeostatic turnover in these antigen-specific memory CD8 T cells in vivo? How fast do they turn over? What is going on in terms of during the maintenance phase and also early on? Okay. Second question we asked is what transcriptional epigenetic changes happen during this thing by analyzing longitudinally in the same individual. Actually, I'm not going to show you this data for lack of time. But then at the end, I'll address some issues about what is about the lineage relationship between effector cells and memory cells. Okay? So there are 2 parts to this talk. The first is this, and the second one is this. So to do this study, we have been collaborating with Mark Hellerstein, who actually is from here. I don't know if Mark or somebody from Mark's lab is in the audience, okay, who was here at Berkeley, but now he has Berkeley and UCSF. And Mark Hellerstein, many years ago, developed this very nice technique of heavy water. So the vaccinees drink, in our case, the vaccinees will drink heavy water for 2 weeks. They will get the vaccine and start drinking heavy water for 2 weeks. Heavy water has deuterium in it, so all the cells that divide will incorporate deuterium. Okay? And then you can follow the deuterium label for extended periods. So the die away, so as you lose the deuterium, only way you lose deuterium is if the cells then divide. Okay? So after 2 weeks, there's no more deuterium in the system. Actually, it takes another 2 weeks for it to wash out. But after like day 30 or so, there's no deuterium in the system. And so now any cell that divides will start losing deuterium. So it's a very nice way of quantitating turnover by this die-away kinetics. So here is the experimental design. And in the next slide, I'll show you the data. And this is actually from the vaccinees that were in the study. Okay? And they were followed out to up to about day 400. So the deuterium intake was over here. So basically, every cell now that is dividing will incorporate deuterium. Okay? And the way the analysis is done is Ramai Kondi, a postdoc in the lab who's done these studies in collaboration with Mark, will sort the tetramer-positive cells. So tetramer-positive cells are sorted at these different time points, 14, 21, 28, so on, up to a year or even longer. Okay? And then we send this to Mark Hellerstein's lab, and then by gas chromatography, mass spec, they quantitate the amount of deuterium in the antigen-specific cells. The day 14 time point is taken as 100% deuterium incorporation, and then after that, we look at the die-away kinetics. Okay? And as I mentioned to you before, of course, a lot of cells are dying, but we're not analyzing the dead cells. Okay? When you analyze the live cell, the only way it loses deuterium is if it divides. Okay? So one can do mathematical calculations that a modeler who worked with Mark Hellerstein has done, and that gives you the estimate of the die-away. So how often do you think the memory cells turn over? Louis, how often? 2 weeks? 1 month? 1 month?
Variable.
Come on. This is antigen-specific cells, Louis. These are not subsets, okay? A lot. All right. You're wrong. They are remarkably quiescent. And the turnover rate from our estimates, and I'll show you a couple of slides, is about 450 days. 450 days. So basically, once you've generated that effector cell there, many of them die, but the ones that survive are amazingly quiescent. They're just sitting there without any turnover, and we can follow them out to about 2 years. And that's the data here. Okay? So this is at the day 14, 100%. So that amount of deuterium is 100% in the DNA of the, again, everything here is sorting tetramer-positive cells of the same specificity. Okay? And this is now the decay. So even out in this few instances, this is out to 2 years, 7 days, 750, and about 50% of the label is still there. Okay? Essentially, you can track a cell that you labeled 2 years later. Okay? It is still there. It has lost less than 50% or so of its deuterium in terms of its specific activity in the DNA. But you'll notice that there's a little bit of a noise over here, a little bit more decline and then a little bit more. There's an aspect of biphasicness in this thing, a little bit more play early on. Okay? So we said, well, by viremia, of course, antigen is gone here, but certainly some tissues will have some. Okay? So we said, well, let's do this experiment. That is, instead of now they drinking water 0 to 14, they drink water 14 to 28, okay, in the next window. And this is the data from these individuals into the tetramer. Again, it's a different set. Okay? And we did the same analysis. And now that biphasic nature is gone. Okay? Because that little bit of tickling they were getting in some tissues, that's gone. Okay? So now it's pretty flat. Okay? Well, of course, it's not flat. It's going to slowly decline, but that little bit biphasicness is gone. Then the third experiment we did was, what will we get if we label during the memory phase? This was all, this analysis was die-away kinetics. You label, and you see how quickly it disappears. The next one was incorporation. That is, you have people who are vaccinated, have memory cells, have them drink the water, and see how much they incorporate. Okay? And the first studies we did here were planned for them drinking for about 30 days. Okay? Basically, if you do 30 days, there's very little incorporation. Okay? So actually, we had to extend it, and they drank the heavy water for about 60 days. Okay? So here in this case, so 100 days or 150 days post-vaccination, these individuals drank heavy water for about 56 days, and then the same analysis was done as we've done before. This time we're looking at not die-away, but incorporation. And very, very low incorporation, very low, if any. And if you, in these studies, we also did the more cruder experiment, which is we just looked at everybody's favorite memory cell subsets, the TCM, TEM, TEMRA, and so on. And of course, there's much more noise over here because we don't know the specificity, we don't know who's coming, who's going. So of course, there is a little bit more noise there. And actually, in the first study that Mark Hellerstein had done, that's what he had looked at. He had looked at HIV-infected people, and he had looked at bulk subsets. Okay? And therefore, there was much more activity going on. But here you see that very little incorporation of these cells. In fact, they're the closest to naive cells. Okay? So they're very, very quiescent in terms of their turnover rate. So what I've shown you so far is the biopsies from memory CD T cells. This is an important point. They originate during the acute phase of infection. This is a key point. The people have argued, when do the memory cells come up? Are they coming at some later time being generated? No, because the only time the label was there was in the beginning, and they still have the label. Okay? So we know that cell that we are looking at 2 years later was generated during this first 14 days. Okay? And again, this is what proves that, because we can track it. And as I mentioned to you, they're dividing only once in about 450 days or so. Okay? Now, I'm not showing the gene expression data, but if you do that or you look at phenotypic markers in the blood, the markers change dramatically. Okay? This doesn't prove it's the same cell that's doing it, because there are different populations, could be there, some cells are dying. But if you do gene expression profiling, effector stage, many genes are up, memory states are down, some which were down are coming up. Epigenetically, we have done analysis of whole genome methylation, and that's changing also. Okay? But what I want to share with you today is data we have on people who were vaccinated about 10 years, 8 to 10 years. Okay? And what is the phenotype of these very late memory cells? Okay? So this is 8 to 10 years post-vaccination. Okay? And again, they're from our same cohort. They're not from the cohort that drank the heavy water, but they're from, actually, I think a couple of them are, okay? But most of them are just people that we had vaccinated before. And what we found here was that these cells, I'll tell you the results and then show you the data. These cells, looking at this 8 to 10 year post-vaccination, look very much like naive cells in terms of their phenotype. And in fact, what they resemble are what several people have described. Work done by Nick Restifo, Luca Guattoni, Mario, what they've described as this TSCM cells. What that means is stem memory cells. I don't know what that term means, but basically that's what they call it. They call them stem. I thought all memory cells were stem cells because they can generate, proliferate, but their new name was put on this population, okay? Which looks very much like a naive cell. And that's basically what the phenotype of these cells is. Okay? So if you now look at, so these are, again, tetramer-positive cells, 8 to 10 years, 12 years after vaccination. They're all CD45RA positive. They're all CCR7 positive. They're CD127 positive, CD28 positive. They don't express any granzyme B. Okay? Of course, that, not surprising. But again, these markers are very much like a naive cell. Okay? And there was a recent paper that came out from a group in Lausanne where actually they scooped us on this thing. Okay? And their data is actually even better. They have even more people were analyzed who got the yellow fever vaccine, and they find the same thing. Okay? That again, these look very much like naive cells. Okay? But of course, they're not naive cells. They do differ in other markers, and I think this is some of the markers that actually I think Lewis uses in the monkeys. Okay? So they have, so this is now looking either at the antigen-specific cells, 8 to 12 years after vaccination, or naive CD8 T cells in that same individual. There's slightly higher FAS expression. And I think the monkey people have been doing this for a long time. So FAS is a very good marker to distinguish naive and memory cells. So the TSCM can be distinguished on that basis. Also slightly higher levels of CD11A. CD31 is lower than naive cells. Again, a good marker to distinguish this TSCM from from naive cells, and they express high levels of CXCR3. Okay. So we then did, or I should say Rama then did what I would say was a very nice experiment. So this was comparing bulk naive cells with the yellow fever specific memory cells 10 years later. But then Rama also got yellow fever specific cells of the same specificity from an uninfected individual. So we are now looking at the naive cells of that same specificity. This is very hard to isolate. You need a large blood volume, but you can do this by tetra enrichment technique. Okay? And this is showing now over here in the light blue are the naive cells of